different devices for different purposes, like camera and video camera, and usually use cell phone just for making calls actually. Yeah, okay, so let's go. Yeah, that's kind of interview. I mean, I want to just cut it into several pieces and show to students in Moscow State University. Yeah, so this kind of maybe a little bit popular. So let's start with the introduction to nanoparticles and this fascinating world. So the first question is, why do we need nanoparticles in biomedicine?
So nanoparticles have been introduced into this field of nanomedicine mainly because we're trying to make improvements in our ability to deliver drugs or deliver better diagnostic tools.
Mm-hmm.
Nanoparticles date back to 20 years where one of the first nanomedicines was an anti-cancer agent called Doxil, which is a nanoparticle that was delivering a molecule that was not very soluble, so you couldn't just inject it directly into a patient's bloodstream.
Mm-hmm.
It was also a very toxic anti-cancer agent, so if you did get it into the bloodstream, it could go elsewhere in the body, not to the cancer, and then cause problems. So Doxil was so successful because it had a therapeutic that was encapsulated in a nanoparticle. The nanoparticle was able to circulate through the bloodstream and focus and localize just on specific tumor tissues.
Mm-hmm.
So then when it got to the tumor tissues, it would release the drug. And so kind of two factors there. One was it was able to make the drug what we call more bioavailable. The drug was insoluble, but it was encapsulated in a nanoparticle, so it was still able to make its way to the tumor.
Mm-hmm.
And the second was it allowed the more selective delivery of the drug, so the drug didn't go everywhere in the body. It was just a little bit more focused on the tumor.
Okay, I'll just let me check if it's okay. Yeah, looks good. So, so the next question is, why inorganic nanoparticles?
Yeah, that's a good question. So why inorganic nanoparticles? Totally biologic nanoparticles are also being used in nanomedicine. Some of the most common ones would be viruses. A virus is a natural nanoparticle, and it can be quite effective for delivery of genes, for example. Inorganic nanoparticles have the advantage that they are more or less a foreign object. The body doesn't recognize it often as say a foreign protein. So one of the advantages of nanoparticles that are from inorganic materials is they may be less likely to induce an allergic reaction in a patient.
Mm-hmm.
That's not always the case, but that's one of the motivation. The second is because they're totally non-natural materials. They don't have necessarily biological consequences that you might find from a biological agent like a virus, for example. A virus may be able to replicate inside the body. That might not be so good.
Mm-hmm.
Inorganic materials also, so they don't replicate, that's one thing, but also inorganic materials don't have, typically degrade into fairly small components that are water soluble. So the types of nanomaterials we work with are silicon-based. They degrade to orthosilicic acid or a silicate-like ion that your body already has in its bloodstream, and so it's a fairly innocuous degradation product.
Mm-hmm.
So those are kind of some of the more common motivations that people use for working with inorganic materials. Another one is that just inorganic materials can have properties that you can't get from a biological material. For example, magnetic resonance enhancement. Iron oxide nanoparticles are used as contrast agents in MRI imaging. If you try to do that same thing with most biologics are not paramagnetic, and so there just aren't any materials out there that can serve that same purpose. So generally, when you look at contrast agents, they all tend to be inorganic in nature, either gadolinium or iron oxide based, or some other more exotic metal based systems.
Mm-hmm. can you tell about like current status of nanotechnology? What do we have here? Is like proof of concept,
Yeah.
or
That's a good question. So what is the current status of nanotechnology in medicine? You know, in the early days, in nanomedicine, we focused on treating diseases that were so desperately bad that a patient was going to die right away, and so anything you could do to help them would be an advantage. So using a nanomaterial that may not be very safe or may have long-term toxicity consequences is not a problem for a patient if they naturally are only expected to live for another 3 or 6 months. And so that's where kind of nanotechnology got its foothold, and that then established some of the safety profiles that we now know for nanomaterials to have. And it also established some of the hazards that we know about nanomaterials now that allow you to figure out where not to use them. And so the trend now, you're seeing that nanomaterials are tending to migrate into other diseases that are not necessarily so dire. Of course, cancer is still a very big theme, but you also see diseases that are not necessarily life-threatening. Ophthalmic diseases where you're just trying to improve the vision of a patient who might be going blind, try to avoid that consequence of them going blind by giving them something in a nanotherapeutic. So without having established 20 years of safety with various nanomaterials, that would have been really hard to do. But so now nanomedicine is kind of at that stage where it's capable of moving into other disease areas, and even health areas like the field of nutraceuticals is really commonly looked at now with nanomaterials.
Thank you. So the next question is about formation of nanoparticles. So we have two approaches, like bottom up and top down.
Yeah.
So can you compare what's the advantages and drawbacks?
Yeah. So the bottom up, so-called bottom up synthesis, means that you're taking molecules or even atoms and putting them together and building some nanostructure. And top down usually means you have some solid, in our case it'd be like silicon, and you carve it out until you make a nanostructure. And there are advantages and disadvantages to both of those approaches, and I think it really depends on the application area that you're talking about. Generally, the top-down approaches, especially if you're working with a material like silicon, you tend to work with a very expensive starting material, and so that limits you to what kind of applications you're going to look at. A top-down synthesis of silicon nanoparticles, for example, would not be appropriate for a battery application where you need to have the material cost maybe pennies or maybe a few dollars per kilogram, but very cheap. Whereas for medicine applications, typical therapeutic, using the example of the eye again, therapeutics that are used in the eye are several $1 million per kilogram. And so when you have a nanomaterial that's maybe only $1,000 or $100,000 a kilogram, it sounds like a lot of money, but it's actually only a small fraction of the total cost of the therapeutic, so it's not a big deal. So then that's where you might use a top-down approach that might be a more expensive process. Bottom-up procedures, they can suffer from problems of reproducibility. That's one of the most common issues with nanotechnology. As you build a structure, especially a nanostructure, it may be different if you change labs or if you change your precursors or temperature a little bit in making the materials. That's true for all of the nano field. Characterization of the materials is really critical. But also from the bottom up, you tend to have more issues with impurities being incorporated into the material from the synthetic chemicals that are used to make them, or just from the feedstock. In the silicon world, we have a part per billion impurities, and silicon is considered catastrophic for the semiconductor industry. And so they've tuned the technology to make really, really pure material. In fact, a silicon wafer is so much more pure than you need it to be for a medicine application. That's not necessarily true with silicates that are derived from other sources. You'd have to spend more money to purify them to make them suitable for medical applications. But overall, it just ends up in what you want to get at the end. And if you've got a certain property, the only way you can get it is from a top-down approach, and that's what you use. If you need to use a bottom-up approach, because you get better properties with that approach, and then people use that.
Okay. So you started to explain a little bit about that topic, but anyway, so why do you work with silicon nanoparticles? What's the advantages of silicon?
Yeah, so silicon nanoparticles have a few advantages. One, they've been pretty much proven safe now in a variety of different application areas. They tend to degrade to non-toxic byproducts. We do a lot of work with quantum dots, and silicon quantum dots are really the only, what we would consider non-toxic quantum dot. Most quantum dots are made out of cadmium or indium or sulfur, selenium components that are reasonably toxic to the liver. So you're looking at using them in the body, that's a real concern. Even if you're going to use this nanomaterial in a lighting application or in a battery or something that you can package it up, and if it's a toxic material, it's not such a big deal. We put lead in our batteries and car batteries, and it's not a problem. Except when the package breaks.
It doesn't explode in there.
And so that's really one of the motivations for silicon, is we're starting with a very low toxicity material.
Mm-hmm. And what's the advantages over silica or iron oxide? Because iron oxide also can be
dissolved. So again, the advantages, not necessarily advantages, but there's different things you can do with the silicon you can't do with, say, iron oxide or silica. One of the chief areas of interest for us with silicon is that it's a semiconductor, so it has some very unique electronic properties that can be harnessed for various biomedical applications. The photoluminescence is this classic one. The material's nice and photoluminescent. You can use it as an imaging agent. You can track it and tell where it is in the body. You can put it underneath the skin, and you can probe it with a near-infrared light to tell whether it's there or it's gone away. Once it dissolves away, the luminescence goes away. And it's a non-toxic source of luminescence. We don't have to use a chemical dye to make it luminesce. So that's one of the areas. Other areas, it's a conducting material. It's photoactive, so it responds to light. There's a lot of interesting aspects that just have to do with the semiconducting nature of the material that you don't see in silica-type systems.
Mm-hmm. Why we need polymeric coating? Because usually we make coating for nanoparticles.
Yeah.
So why do we need it for biomedical applications?
Yeah, for any nanoparticle that goes into the body is going to get coated with proteins. And those proteins are going to come out of the body naturally as part of the body's defense mechanism for eliminating foreign objects from the bloodstream. And so coatings are an attempt to typically fool the liver and the other organs and mechanisms in the body that are designed to take those nanoparticles out of circulation, to allow those particles to circulate longer and then deliver the therapeutic or image the tumor that we're trying to do in our experiment. So coatings are really important for that, so-called biocompatibility, which really in nanoparticles generally means that it's still going to get coated with proteins, but those proteins aren't going to trigger an immune response that causes the body to eliminate them right away.
Mm-hmm. So we're going to biocompatibility and toxicity issue, and can you split all nanoparticles into two groups, like toxic and non-toxic? I mean that's relative, but
it would be very easy to. It'd be great if you could say split materials into a toxic and non-toxic categories. In fact, there are many materials that are highly toxic in one domain but totally safe in another domain. And so toxicity is all about the dose and the availability. So there are a lot of materials. You just think about alcohol. If you drink a lot of alcohol very quickly, you could die.
Yeah.
But if you drink it slowly, you may not even have an effect. And so if you give a very small amount, and you won't even notice it. So that's, ethanol is considered a toxin. It's very toxic to cells in certain concentrations. Mm-hmm. You keep it down low, then it's not a problem. And that's true with any material. Generally, though, the trick is to try to have this material eventually go away. And so we talk about self-destructing nanomaterials. If it's something that doesn't degrade under any bodily conditions or in the environment, unless you go to super high temperatures or very corrosive conditions, it's probably not a, you have to be careful of how you use that nanomaterial. You have much more limited applicability of that kind of material, because if it doesn't go away, it'll eventually cause some problem. And you look at asbestos, which is a classic example of a nanomaterial that's very dangerous. If you breathe it in, it'll give you a cancer, 20 years later. But it's used in ceiling tiles to keep houses from burning down. So actually it was a very safe material as long as you don't breathe it in.
Yeah.
And it's a natural mineral. You can dig it out of the ground. So, really, really depends on how you're using it and how much of it you're using.
Okay, but can you say what is the best biocompatible nanoparticle, or there's no easy answer?
Yeah, there's no easy answer to that, but probably the best biocompatible nanoparticle is one that the body already knows how to get rid of. And so that's really where the natural nanoparticles tend to have an advantage. So you look at viruses, I've already mentioned, that's a natural nanoparticle. And a virus, of course, can be very harmful. If it's a cold or flu virus, it gets you sick. There are other viruses that are quite harmless, and so the body knows how to deal with them. And even if you do catch a cold with a virus, your body will eventually build up an immune response to deal with it. And so in that regard, biological nanomaterials have a benefit in that the body has mechanisms that can degrade biologic systems. And inorganic systems, they have to be programmed to degrade, in their design. If they don't, there may not be any biological mechanisms that will degrade them.
Mm-hmm. So size is really important for nanotechnology. I mean, it's all about size, actually.
Right.
And so what's the best size for, for like intravenous injection?
Yeah.
Or
So in nanomedical applications, so again, there's lots of different ways to use nanomedicines. You can apply them topically on the skin. But let's just talk about intravenous administration. For IV administration, if a nanoparticle's smaller than 5 nanometers, the kidneys will filter it out. So generally, if you have something that's 5 nanometers or smaller that's circulating in the bloodstream, the elimination pathway will ultimately be through the urine, into the kidneys and out through the urine. For something larger than that, then usually the spleen or the liver will take that nanoparticle up and pick it up in called Kupffer cells inside the liver and eventually get excreted into the bile. So bigger particles tend to be eliminated a little bit more slowly until you get really big. So there's kind of a sweet spot. We talk about working between sort of 10 nanometer at the small size and up to maybe 200 nanometers on the large side. You start getting larger than 200 or 300 nanometers, then the nanoparticles, as they're so big, they're going to start getting filtered out much more effectively by the various processes the body has to deal with larger objects. They're too small, then they just act like small molecules and they get excreted just like they would, like any small molecule would.
mm-hmm.
Again, that's assuming that you want it to circulate for a long time. But there are some applications where your nanoparticle, it needs to go in and do a quick job of imaging, let's say, and then just get right out. Maybe you only want it to circulate for 10 minutes or for 2 hours, and you maybe don't necessarily have to worry about those size issues.
Mm-hmm. By modification of the surface, we can change the biodistribution, right, of the nanoparticles in the body. So, do we need it? I mean, can we use like different biodistribution for different particles, or we need just to like pick up the best way and make all the particles excreted in the same way?
Yeah. I guess that maybe you could restate the question. I'm not sure exactly what you're.
I mean, we can play with surface composition, right? And like put the particles into different excretion way, like through liver or through urine.
Yeah.
So I mean, is it helpful, or we just need to like find the best way and put all the particles into that way, just to get rid of them.
Well, generally the surface chemistry kinds of issues fall into various categories, but one of them is charge. So if you put a lot of positive charge on a nanoparticle, it's now pretty well accepted that very highly positive charged particles are going to denature proteins. So keep in mind that anything that goes in the bloodstream is going to get coated with protein. And you have these cells called scavenger receptors on cells that are called scavenger receptors. And those receptors will recognize denatured or aberrant proteins and pull them out. So if you take, say, any given nanoparticle and you put a chemistry on the surface that makes it positive, it doesn't matter so much what's inside there, but that positive surface charge will attract proteins, and those proteins, if it's high enough charge, those proteins will get denatured, and that'll then activate the scavenger receptor pathway. If they're negatively charged, the chemistry can be identical except for that. The proteins will still coat that surface, but those proteins may not get denatured, they may not get recognized. But then it really depends on what proteins stick there, because many proteins, even if they're not denatured, they can trigger an immune response or phagocytic response that would cause the particles to be taken up. And so that's kind of the game we always play, like what's the best chemistry, and some of it's, you just have to figure it out phenomenologically. But what's happened in the end is people have kind of come down to certain types of chemistries that seem to be good at keeping particles from getting removed from circulation too quickly. Polyethylene glycol is a big one. Dextran, it's a sugar, is another one that's used a lot for nanoparticle coatings. In both those cases, you're looking at surface charge is pretty much neutral. If you go too far negative or too far positive, they tend to get taken up pretty quickly. If you make the particles neutral, then you might have a problem getting them to stay suspended in solution, because neutral particles will tend to aggregate and fall out of solution. And so that's the trade-off that people play with, and how they design their structures to keep them stable in a vial, but then also stable long enough in circulation.
Mm-hmm. Okay, so let's move to applications. Of course, we have a lot of promising applications. So if we compare like in vitro and in vivo methods, what do you think, what's the most promising for applications of nanotechnology?
Yeah. Yeah, in vitro versus in vivo is a good separation point. When you talk about in vitro nanotechnology, you're doing an experiment in a petri dish. Maybe you're trying to identify a certain cellular receptor or identify certain types of cells in a large assembly of cells in a dish. Or you're trying to identify a certain function in those cells. You can do whatever you want with that nanoparticle, and it can be highly toxic. As long as it's not so toxic that cells die immediately. If it's something that's sort of toxic, so after a week or so they would have a problem, and you can get away with that. So the designs are really relaxed for any kind of an in vitro experiment, if you're just trying to develop that nanotechnology to help you learn more about the biology happening in a cell. If you're trying to make a nanomedicine, that's where you have to start thinking about the longer term consequences of the material, whether it's stable, whether it's safe, the toxicity issue becomes important. And again, so in vivo, the biggest issue is toxicity. Once you've figured out that it actually works, so let's say you want to do drug delivery, so you got a nanomaterial and it loads the drug that you care about, and it loads it in some reasonable level. Okay, then the next question you got to ask is, will it be safe? First, it kind of has to have the function, and then you have to figure out whether it's going to be safe. In an in vitro experiment, you don't care about safety so much.
Mm-hmm.
The main thing then you're concerned about is, will this nanomaterial interfere with the biology? So I'm trying to study some, let's say a cellular uptake process with my nanomaterial. Well, will the nanoparticle itself induce some changes in the cell that causes them to act differently, and you're learning something that you don't realize what you're learning in that experiment, and maybe something that's more specific to the nanoparticle and not something that's more generally applicable in biology.
Mm-hmm. Okay. So we have a lot of, basically we use nanoparticles as contrast agents for different methods. So what do you think for which method is like the most beneficial? Which method requires really new contrast agents?
Yeah, the contrast agents, fall into two different camps in a way that if you have a deep tissue imaging agents, kind of like magnetic iron oxide nanoparticles or gadolinium systems, they're used to enhance magnetic resonance imaging, or you might have agents that enhance PET, positron emission tomography. Those are techniques that look deep into the body, and so those agents are meant to enhance the contrast of those imaging tools. If you're looking at fluorescence, you're not probably going to look at deep in the body. People try really hard now to make nanomaterials that are fluorescing in the near infrared so that you can see them deeper in the tissues, but they always tend to be pretty near surface types of probes. So where a fluorescent nanoparticle may be useful would be what we call like an intraoperative procedure. So a doctor's cutting a patient open, they're cutting out the tumor, and they don't want to go back in and have to cut it out again. So they want to see what we call the tumor margins. And so while the doc is in there, cutting out a tumor in a patient, they want to have some imaging agent that just shows them where that tumor is and where the healthy tissue begins. Mm-hmm. And so they can cut out just the stuff. And so there, if you have a fluorescent nanoparticle, then you can put on the right glasses, put it under a black light or whatever, and then the doc in real time can see the disease cells and so cut them out better. So that's usually where you see the fluorescence kinds of applications, if they're going to translate to the clinic, if they're not just going to be used as a tool to understand biology better. But there are other application areas, where near surface is okay. So like I just described one in a surgical procedure. Another one would be, something that's a transdermal patch or a patch that's used to deliver a drug, or maybe a subcutaneous implant that's delivering a drug to a patient who, maybe a chronic pain medication. And you might want to just look through the skin to see if that fixture is still there or how much longer it has to go before you need to change it out and give the patient a new fixture. And there you could use something like fluorescence as a probe.
Mm-hmm. Can we use nanoparticles for screening? I mean, for like apply for every healthy person in the world, for make sure that everything is good.
Yeah, that's the difference between therapeutic and diagnostic, is what you hit on. So therapeutic is something where you're treating a sick patient, you're trying to get them better. So as I mentioned earlier, nanomedicine started in the cancer domain because all you had to do is buy the patient a couple of months sometimes, and it actually would be a very effective therapy. In diagnostics, you're giving these things to healthy patients, and the FDA has set out guidelines and rules about nanomedicines for these two domains. And when you're using it as a diagnostic tool, screening to see whether a patient has a cancer or some other disease, then it's required that material be safely degraded or eliminated in what they call a reasonable amount of time. And usually that means within a few hours after you've done your diagnostic test. Right? You just want it to get out of the body. And that's true of radio labels and lots of other therapeutics or diagnostic systems that are in use. And so in the nanomedicine area, having a material that degrades to something that's harmless is really critical for both those types of applications.
Okay, so we know that nanomaterials is really flexible framework, and we can use like multimodal contrast agents, multimodal imaging. So do you think is it really necessary? Can it really improve the medicine level? To use the same nanoparticle,
yeah. That's been a thread so this idea of multimodal imaging has been a thread in the research community for quite some time now. And Jiho Park, who was a PhD student in my lab many years ago, had a slide he showed at his PhD defense, which was a really good one. There's two pictures, one of a U.S. soldier with like all of his equipment and backpack and gun and everything, and the mask and the helmet, and then the guy next to him was Bruce Lee. It was like wearing just like a little, Speedo. And he said, which one is more deadly? He said, well, it depends. But they're both pretty effective. And his argument was that really nanomedicine should be looking more at the Bruce Lee approach, where you just have it designed to do one thing and do it one thing really well, and not load that fighter up with a bunch of other equipment that they're not going to need necessarily for the task that you need them to do right now.
Mm-hmm.
So, nanomedicine, people say, oh, this is a great tool because it can be imaged in multiple modes. Well, in the end, when a doc says, okay, I want to figure out what this patient is sick, they're going to probably call out one test. I mean, if that one test doesn't work, then maybe they'll call a second test out. But it's never made sense to me why you would want to have a nanomaterial do too much. I think I really am much more in favor of this sort of Bruce Lee nanomaterial approach.
Mm-hmm. Okay, thank you. And let's move to therapy. So you mentioned already that it all started with oncology and anti-cancer treatment. So what do you think? Can it make it really different in cancer treatment? So will we see nanotechnology everywhere in oncology?
Yeah, I think what a lot of the lay public and don't maybe realize that nanotechnology is already pretty ubiquitous in the world, and not only in the field of medicine, but Gore-Tex, which is a cloth that you wear in your jacket. It's a water repellent cloth that has 100 nanometer pores in it to keep the liquid water from going through, but those small pores let water vapor exit, so you wear it and you don't get all sweaty on the inside, but you don't get wet when it rains. That's a nanomaterial, and that's been around for 30 years. And so I say, oh, is it a safe material? Well, it's pretty safe. And in nanomedicine and cancer treatment, we're already using these things. In fact, Doxil I mentioned earlier is so old, it's now off patent. I mean, the original patent on that nanomedicine is now expired, and you can buy generic Doxil. So there are a number of nanotherapeutics that are already approved and in use, and there are a lot more that are in the pipeline that people have submitted, or they're about to be released. And so we're going to see more and more of that. And the bottom line is because it improves the efficacy of our therapeutics, it makes them work better.
Mm-hmm. Okay. And what's the future of targeted drug delivery? Because that idea came up like decades ago, and I mean, we don't see a lot of targeted drug delivery drugs
right.
in the stores. So how can we improve the drug delivery, or if we're gonna use active or passive targeting?
Right. And targeted delivery, was one of these ideas that was really great on paper, but in implementation, sometimes it works and sometimes it doesn't. And there are a lot of reasons for that. One of them, which I think is a big misnomer in the community, is just because you have a targeting group on your nanoparticle doesn't mean it's going to target. So if that targeting group gets coated with a protein, it never makes it to its target. If the cell receptor that you're targeting is very low expression, has a very low expression level, you can easily saturate that receptor. And so you throw in a little bit of nanomedicine, and the nanoparticle hits that receptor and sticks to it, and now you've saturated all your receptors. If you double your dose or 10x your dose, it's not going to be any more effective because you've already saturated the receptors. So this issue of receptor saturation is really critical in the targeted nanomedicine field. And there are a lot of people who don't really appreciate that because they just say, well, the cells express this thing, we're going to use it. And then they'll do an experiment in a petri dish and they'll say, yeah, it really works effectively. And then you go in vivo and it's not as effective. So receptor saturation is an important issue. And of course, that's not an easy question to answer. I mean, it's going to be very specific to whatever disease you're talking about, whatever targeting group you have that you're using. But I think the field is mature enough now that there are effective targeting groups out there that are being translated into the clinic. RGD will probably become, it's already been approved for, actually it's not been approved yet, but I think RGD is already in clinical trials, and some of those clinical trials, if they don't fail, then you'll see it. I think there's a number of other targeting groups that are under development, that once they hit and they find the right disease and the right dose, you'll see that get much better accepted. But currently, I think targeting, it's a mixed bag, in part because sometimes you have a good targeting group and it might be too good, and it then induces, it targets some receptor that also is important in some other part of the body, and so you end up screwing up that mechanism. That that's not really what you're hitting your target, but the target that you're hitting is doing something else biologically that you didn't anticipate when you thought, oh, this is just going to target to the tumor. That's another issue in the targeting domain, is that all cells have different types of chemistries on their surface, but tumor cells are mutated human cells. They're mutations of healthy cells, so they're going to express receptors and things biologically. They're probably pretty close to other cells in your body, and so it's just a question that maybe they're overexpressing certain receptors, not that they have some brand new cancer receptor that no other cell in your body has. And so the sort of magic bullet of being able to use a targeting group just to go after that one cell and ignore all the other cells, it just doesn't make sense from the biology of cancer. You can at least improve it, so if you can get that nanotherapeutic to the tumor twice as effectively, sometimes that's enough. You drop your dose down, you still are effective at killing the tumor without being so toxic to the patient. That's an improvement. But it's incremental. It's not necessarily a world-changing.
Okay.
I think there are world-changing applications out there for targeted therapeutics, and they just haven't been identified yet.
Okay, so we have to wait a little bit.
We'll see.
Okay, so another idea is so-called triggered release, when you load drugs and deliver, and then simultaneously unload it. So what do you think about this idea?
Oh, triggered, yeah, so generally triggered release is a really powerful and important concept. It's still very early stage, and identifying what kind of triggered systems can be most effective is really critical in that. Simple triggering based on pH is already out there. I mean, you look at enteric coatings on pills that you take. Sometimes those pills are designed with a coating that can't dissolve in the stomach but does dissolve down in the lower intestines, because that's where the therapeutic's more effective. So that's basically a triggered response. You have even just a simple thing as a chemistry of a coating that dissolves at different spots in the GI tract is established, and it works well for some kinds of therapeutics. So you're not going to, the concept's already out there, and I think it's a good one. It's going to be used. Where it hasn't seen as much success yet is in the, in vivo, and in circulation, where the therapeutic that you're delivering, maybe it's triggered by a certain enzyme that's overexpressed in the vicinity of a cancer cell, or it's triggered by a certain difference in pH near, infected cells or inflamed cells. But I think that's going to come. That's the sort of chemically triggered concept that I'm talking about. There are these other triggered therapeutics, which we refer to as externally triggered therapeutics, such as maybe a magnetic material that when you put it into you give it to the patient, and you put the patient in a magnetic field or an oscillating radio frequency field, that the particle moves faster and heats up, and it releases its payload. So you could trigger that from an external stimulus, or a flash of light, or some kind of high intensity ultrasound pulse to cause the nanotherapeutic to be triggered or responsive. Those are even more poorly developed at this stage. Right.
Okay, that's a good point for our research.
Yep.
And another idea is personalized personalized medicine.
Yeah.
People talk a lot about this, so how can nanoparticles help to this concept? I think they should be...
Yeah, I think, it's just, there's no specific, nanomedicine will have a role to play in personalized medicine, just like it'll have a role to play in any, even in non-personalized medicine. The idea of personalized medicine, where you maybe know something about the genetics of the patient, and they're going to respond more effectively to this kind of drug or that kind of drug. Yeah, that's what we mean by personalized medicine. Well, if one of those drugs is a nano drug, it just happens to work better on that person genetically, because genetically their body is more, or the tumor they have is more predisposed to being attacked by that particular approach. And yeah, you'll see it. But I don't know if nano has any special role to play in personalized medicine. It'll be a big role, but it'll be true for really even a small molecule therapeutic. If you know which pill they give the patient, you're going to be better able to treat them.
Okay. For other applications, nanoparticles can be used for, like, against